In this video, I want to introduce you to genetic linkage and talk a little bit about mapping in the eukaryotes, and specifically addressing portions of the learning outcomes related to what genetic linkage is and how it affects the outcome of crosses, what is genetic mapping and why it's useful, and how crossing over can change the arrangement of alleles along a chromosome. Now, first, let's talk about genetic mapping. This is also known as gene mapping or chromosome mapping. Its purpose is to determine the linear order of linked genes along the same chromosome. Now, here's a genetic map for flies, actually. And you can see where there's different genes that have been identified, such as the L gene that controls the size of the animal. Here we've got a gray body, with the ebony gene that controls the color of the body. Sorry, I should point out that here this is listed. You'll see the wild type phenotype and the mutant phenotype. In the flies, the mutant phenotype is used to actually name the genes, while the wild type phenotype is what's most prevalent in the populations. So what you'll notice here that each gene has its own unique locus at a particular site within a chromosome. And that's standard for all maps that are found in animals or other organisms. So these genetic maps can be really useful. They allow us to understand the overall complexity and genetic organization of a particular species. They help molecular geneticists to clone genes because they know the location of these genes. And they can improve our understanding of the evolutionary relationships between different species. They're also used, the maps themselves, to diagnose and perhaps someday to treat inherited human diseases. So if you look at a genetic map of human chromosomes, they can help in predicting the likelihood that a couple will produce children with certain inherited diseases, and provide helpful information for improving agriculturally important strains of other animals through selective breeding programs. So first I want to talk about the distinction of linkage, i.e. whether it's physical versus genetic. So each species of organism must contain hundreds to thousands of genes, yet most species have at most a few dozen chromosomes. Therefore, each chromosome is likely to carry many hundred or even thousands of those genes, and the transmissions of genes close to one another or on the same chromosome typically will violate Mendel's law of independent assortment. That is all based on the distance. So there's two terms that I want to point out here, and that's synteny, and that's the concept where two or more genes are located on the same chromosome and are physically linked. So they're linked by being present on the same chromosome. Now, genetic linkage is a little bit different in the sense that in this situation, the genes are so close together that on that chromosome that they tend to be transmitted as a unit. Now, that then can violate our assumptions regarding independent assortment and the probability of seeing certain phenotypes in organisms or offspring. Now, chromosomes themselves are referred to as linkage groups. That means they contain a group of genes that are linked together. And that we use this term of linkage group. It's based on the number of types of chromosomes of a species. So if we think about humans, how many linkage groups do humans possess? Now, in general, there are 22 autosomal linkage groups, because we possess 22 autosomal chromosomes. Then we also have the sex chromosomes. So the X chromosome represents a linkage group. The Y chromosome is a separate linkage group also. So technically an individual that's a female would have 23 linkage groups, and a male would have 24 linkage groups because they have both an X and a Y. Now, genes that are really far apart on the same chromosome can appear to independently assort from each other. And this is typically due to crossing over or recombination, if you think about it. Now, there's two different kinds of crosses that are typically used to study linkage between genes, and simply You could refer to a two-factor cross when you're looking at two genes, three-factor crosses when you're looking at three genes. Now, the three-factor crosses are kind of cool because that allows you to determine the order of the two genes. If you have, of the three genes. If you have just two genes, A and B, you don't know if A is on this side of B or if it's located on this side. If you throw in a third gene, then you can identify their position relative to each other. Now, Basin and Punnett were working with two traits, and they noticed something. Well, these traits weren't assorting independently. It suggested that the transmission of these two traits from the parents was somehow coupled. They did not realize that the coupling was due to the linkage of the two genes on the same chromosome. They didn't have that ability to detect that. So what we see here are purple flowers and red flowers. And then these purple flowers also have long pollen, and these red flowers have round pollen. So there are two traits, our two characters are the color of the flower and then the shape of the pollen. Now, when they mated these two true breeders for their F1, they got all heterozygotes again. However, when they mated their heterozygotes from the F1 generation, they got this F2 generation. Now, under our assumption of independent assortment with a dihybrid cross, we should expect this ratio of 9 to 3 to 3 to 1. And these would be the expected numbers based on the total number of individuals that they observed in their population. Now, what you'll notice here is that this class, this phenotypic class, and this second phenotypic class, which represent the phenotypes of the parents, are actually in a higher frequency than you would expect under independent assortment. These two other classes here exhibit a lower number of individuals, much lower than what you would expect under independent assortment. 19 versus 80, 27 versus 80 again. Now, this caused them to think something was going on. Now, crossing over in general can produce what we refer to as recombinant phenotypes. So that kind of phenomenon actually causes linkage groups to be altered during meiosis. And remember that crossing over is going to occur during prophase 1 of meiosis. And this is that point where we're actually seeing these bivalents lining up on that metaphase plate. So any kind of crossing over that we're talking about is occurring between the non-sister chromatids of homologous chromosomes. And that is a true exchange of DNA segments. So let's look at this a little bit more specifically. So here we've got our diploid cell after replication. We've got a pair of homologous chromosomes lining up and forming that bivalent. If there's no crossing over, these linked alleles are going to segregate together, such that basically you've got big A and little a together here, and then in another gamete you've got big A and little a, which is coming from that homologous chromosome, and then alternatively the sister chromatids of this dyad are separating to form these two. So in this situation, we're assuming that these are so close together that there's no crossing over that's happening. So this violates our independent assortment, because under independent assortment, we would expect equal probabilities of each of these gametes and equal probabilities of all the possible combinations. So the haploid cells are containing the same combination of alleles as the original chromosomes. Now, in this situation, we get crossing over. We've got her starting out still with our bivalent. Synapses occurred, and we see all the possible combinations of these alleles. This is similar to what we would expect under independent assortment. So it's the same kind of pattern in that sense, but as we will explore further, how we can differentiate independent assortment from linkage and crossing over. So these haploid cells contain a combination of alleles that are not found in the original chromosomes, as you can see, as demonstrated by this haploid gamete and this haploid gamete. These are typically referred to as, these down here, are referred to as non-parentals or recombinant cells or recombinant chromosomes, whereas these are referred to as parentals. Now, in these other videos, we'll talk about how you determine when linkage and crossing over is actually happening in organisms.